Good morning, I'm Dr Ramya, Associate Professor, Department of Physiology, Sri Lakshminarayana Institute of Medical Science. It is affiliated under Bharat Institute of Higher Education and Research. Today, we will be dealing with the physiology topic, gastric secretion. We will move on to the topic now. These are the different parts of the stomach, the function and anatomy. This is the esophagus. The lower end is connected to the stomach. In between them, there is a sphincter, which is the lower esophageal sphincter. This is the cardiac end of the stomach. This is the fundus of the stomach, which is dome shape that is present on the upper part of the stomach. This entire area is the body of the stomach, and it extends into the pylorus, from the pyloric anther, or towards the end of the stomach is the pyloric sphincter. So there are two sphincters, one on either end of the stomach, one on the upper end, which is the lower esophageal sphincter, one on the lower end, which is the pyloric sphincter. So to revise, fundus, this is cardiac end, this is the body of the stomach, and this is the pyloric fracture. This is the lesser curvature of the stomach, and this is the greater curvature of the stomach. So when we go into the physiological aspects of the gastric glands, there are three main types of gastric glands. The first one is the cardiac gland. Then we have oxyntic glands, then pyloric gland. The cardiac gland, as the name suggests, is present in the cardiac region of the stomach. Then there is oxyntic gland, which is present in the fundus and the body of the stomach. Then there is pyloric gland, is present in the pyloric region, that is the pyloric anther. So this is the area where the cardiac glands are present. This is the area where we have the presence of oxyntic glands, and this is the area where we have the presence of pyloric glands. To know this function, you can always correlate with this location. The cardiac glands and the pyloric glands, if you see, their main function is to secrete mucus, which is rich in bicarbonate here. In the same way, pyloric gland, if you see, they also secrete a lot of mucus to mucus cells, and it also has a specialized cell called G cell, which is nothing but a local enteroendocrine cell, which helps in the secretion of gastrin. Why do we need the presence of mucus in the cardiac gland and the pyloric glands? So depending on its location, we have to correlate, I say, because these cardiac glands are present near the cardiac end of the stomach. Pyloric glands near the pyloric end of the stomach. So here, the gastric content may get regurgitated. So this main purpose is to provide mucus to prevent this erosion. The same purpose here at the pylorus also. Then we move on to the other gland, an important gland in the gastric area, which is the oxyntic gland. Oxyntic gland is present in the fundus and the body of the stomach. These glands, they contain a specialized type of cell called oxyntic cell. Oxyntic cell is also called as parietal cell. So apart from this cell, they have cheek cells or peptic cells, mucous neck cells, and endocrine cells. The function of oxyntic cell or the parietic cell is mainly to secrete hydrochloric acid and intrinsic factor. Cheek cell or the peptic cell, as the name suggests, peptic, it is involved in the secretion of the release of pepsinogen. Then mucous neck cell, involved in the release of mucus, thick mucus, which is highly rich in bicarbonate. This is highly important in the gastric secretion because this forms a layer, a mucus bicarbonate layer, which forms a gastric barrier preventing the gastric mucosa from getting eroded by the action of the acidic content of the stomach. So apart from these cells, it also have a local endocrine cell called ECL cell, enterochromaffin-like cell, that secretes histamine. Histamine is a potent stimulator of gastric secretion, especially the HCl secretion from the parenchymal cell. So now we'll move on into the structure of oxyntic gland. This is the structure of the oxyntic gland. We can note down the presence of the mucous neck cell in the isthmus area. Then we have the parietal cell, we have the chief cell and ECL cell. So this is how the parietal cell is located. Can you note that they are present one near to each other all through the gland structure? So the purpose of the mucous neck cell is to secrete mucus, which forms a barrier. Parietal cell or oxyntic cell, they secrete HCl. You can note down here that the apex is facing the lumen, base opposite to that. And this is the chief cell and this is the ECL cell secreting system. So this is how they are. This picture shows the overall function and the structure of these cells in the oxytic gland. See, this is the mucous neck cell, chief cell, parietal cell, their function. So mucous neck cell, it produces an alkaline mucus, that is mucus rich in bicarbonate forming a gastric barrier. Then we have chief cell, whose name is also peptic cell, pepsinogen secretion. Parietal cell, HCl and intrinsic factor. This pepsinogen is converted to its active form pepsin by the action of HCl in the stomach. So, their function, you can have an overview of it. See, intrinsic factor and HCl from parietal cell, pepsinogen from chief cell, then mucosa from the surface cell. I've told you there is a localized cell which is endocrine in nature, G cell, which secretes gastrin, which is located at the pylori. Now we'll move on to the specific structure of the parietal cell. This is how a parietal cell looks like. We can note down this is the apex of the parietal cell and this is the basal end of the parietal cell. So the opening of the cell is located on the apical surface. So these are villus, microvillus structure present on the apical membrane of the parietal cell. And they have a canaliculi through which the secretion gets into the lumen. It contains a lot of mitochondria, which is helpful in the supply of energy in the form of ATP for this movement of hydrogen into the lumen. Then we have lysosomes. Apart from the two important structures like tubules and vesicles are also present in the parietal cells. The hydrogen potassium ATP sperm, which is involved in the secretion of hydrogen into the canaliculus, is incorporated here in tubules and vesicles. So we move on to the composition of gastric secretion. So gastric secretion, if we see the volume of gastric secretion per day, it amounts for about 1,500 to 2,000 milliliters per day. It varies between individuals from the lower range to the upper range of the normal level. The acidity or the pH of the gastric content, it varies between 0.8 to 4. So it can be very acidic as high as with pH of 0.7 or 0.8, depending on the stimulus which is being given from this parietal cell. So overall, if we see, whatever the secretion may be from the gastrointestinal tract, the majority constituent of that particular secretion is mainly by the water, constitute about 99 to 99.5% of water. The remaining by the solids about 0.5%. So in that solid component of the gastric juice, we have organic substances and inorganic substances. Inorganic substances, so we can easily say what is the main inorganic substance present in the gastric secretion? All of us know. Yes, it is hydrochloric acid. So we have hydrogen and chloride in the gastric secretion. So apart from that, it also has sodium, calcium, potassium, bicarbonate, phosphate, and sulfate. Then we move on to the organic constituents. So in the organic constituents, we have enzymes and we have other organic substances. In the enzyme, if you see, the first one you should always remember is about the pepsinogen. So this pepsinogen is secreted from the peptic cell. So which is converted to pepsin by the action of hydrochloric acid in the stomach. Then we also have gastric lipase, gelatinase, and urease. So apart from that, the other organic substances like mucus and intrinsic factors also be a major part of the gastric secretion, because mucus, as we discussed earlier, it forms a thick gastric barrier for about a mm. So sometimes it can be more than a millimeter thick also. It forms a thick mucus layer preventing the gastric mucosa from getting eroded. And there is also secretion of intrinsic factor, which is highly important for the absorption of vitamin B12, which happens in the intestine. Now we move on to the mechanism of HCl secretion as such. So we have already seen in the previous, like one of the previous slides, about the structure of parietal cell. So this is how the cell looks like at resting state, and this is how the cell looks like whenever it is stimulated and it is highly secreting and active HCl. So here, if you see at resting state, so I've told you this is the canalicus of the parietal cell. This is the apical side and this is the basal side. There are a lot of tubulovesicles inside the cell. So these things are nothing but hydrogen potassium ATPase pump, which is mainly responsible for the secretion of hydrogen into the lumen. Okay. This is the structure of the cell at rest. Rest means whenever it is not actively secreting hydrochloric acid in response to any kind of stimulus. Okay. So this picture, if you see on the side of it, is the cell at actively secreting phase. Actively secreting phase in the sense whenever it is actively secreting hydrochloric acid in response to a stimulus. So this is the structure of the parietal cell during active HCl secretion. So in this picture, we can note down that this tubules and the vesicles, which has hydrogen potassium ATPase pump, they go and get fused up with the canalicule on the apical membrane. Can you note down? Yes. So tubules, vesicles, they get fused with the canalicule on the apical membrane. So what happens to the surface area available for the secretion when compared to the resting state? The surface area available for the secretion of hydrochloric acid has increased here, forming a bigger area available for HCl secretion. So we can note down the presence of the hydrogen potassium pump here. So this hydrogen potassium pump gets incorporated into the walls of this tubulovesicular canalicule on the apical surface of the parietal cell. So here, what does this cell do? This hydrogen potassium ATPase pump? This secretes hydrogen in exchange for potassium ion. So the secondary messenger involved in this cell activity is calcium cyclic AMP in case of histamine, calcium in case of acetylcholine. So this is a schematic representation of all those molecular activities. We can note down this is the structure of the parietal cell. This is the luminal side, that is the apical side of the membrane. This is the basal side, interstitial space. So I spoke about hydrogen potassium ATPase pump in the tubulovesicular canalicule membrane. So we can note down the presence of hydrogen potassium ATPase pump here. So how do we get this hydrogen? Where do we get this hydrogen from? Usually inside the cell, as a product of metabolism, we always have carbon dioxide. So apart from that, sometimes the carbon dioxide also diffuses from the plasma to the interstitial space in the cell. So we have carbon dioxide inside the cell, which associates with water inside the cell and forms carbonic acid. So this reaction is catalyzed by the enzyme carbonic anhydrase. So this carbonic acid which is formed again dissociates to form hydrogen and bicarbonate. The hydrogen formed by the dissociation of carbonic acid moves into the lumen exchange for potassium. So this hydrogen has to move into the lumen and potassium has to get into the cell as an exchange process, but this requires a lot of energy. So normally the energy is utilized in the form of ATP. So we can note down the hydrogen concentration on the luminal side is very high when compared to the hydrogen concentration inside the cell or the plasma. So it has to move against concentration gradient. We know about the primary transfer. So no, when something has to move against a concentration gradient, it requires the expenditure of energy. So here ATP is needed for the movement of hydrogen into the lumen in exchange for potassium into the cell. Now hydrogen has moved into the lumen, first into the canalicular pit, then from there into the lumen. So for every hydrogen form, we also have the formation of one bicarbonate ion. What happens to this bicarbonate ion? This bicarbonate is moving out of the cell in exchange for one chloride ion into the cell through chloride bicarbonate exchanger. So this chloride diffuses through the cell and passes into the gastric lumen by means of chloride channel, CFTR channel. So here in the canalicular membrane, if you see, we have the presence of hydrogen and also chloride. Both of them combines with each other to form hydrochloric acid. Okay, so now we have the formation of hydrochloric acid. But what happens to this potassium which is being exchanged for hydrogen? This again is being recycled back into the gastric lumen for this pump to happen. Okay, then the level of sodium and potassium inside the cell is also maintained by sodium potassium ATPase pump on the basal side of the membrane. Okay, so we'll revise this again once. We have carbon dioxide inside the cell as a product of metabolism, and we also have carbon dioxide penetrating from the blood through the IC into the interstitial fluid into the parietal cell. So which combines with water to form carbonic acid in the presence of carbonic anhydrase. Carbonic anhydrase catalyzes this reaction. Then this carbonic anhydrase, this is the formation of carbonic acid. Carbonic acid again dissociates to form one hydrogen and one bicarbonate. This bicarbonate is exchanged with potassium by hydrogen potassium ATPase pump. We need the utilization of energy because this hydrogen is moving against the concentration gradient into the lumen. Then, for every hydrogen form, there is formation of one bicarbonate which is being exchanged with chloride on the basolateral side through chloride bicarbonate exchanger. So, for every hydrogen entering the lumen, one bicarbonate is entering the plasma. This chloride again moves into the lumen through chloride channel, where hydrogen and chloride combines with each other to form hydrochloric acid. This potassium is again recycled back into the gastric lumen through potassium channels. Then the potassium and the sodium concentration inside the cell is being maintained by sodium potassium ATPase pump on the basolateral side. So what happens to this bicarbonate which is moving into the plasma? See, you can know now for every single hydrogen moving into this canaliculi, one bicarbonate is moving into the plasma. So you can know now it is proportionately moving. So for every hydrogen moving this side, one bicarbonate is moving towards the opposite side into the plasma. So whenever there is increase in acidity, there is proportionate increase in the alkalinity of the blood, causing postprandial alkaline type, where the alkalinity of the blood and the urine is high. So when you think about HCl secretion, you have to always remember about these two pumps. Hydrogen potassium ATPase pump, which is also called as proton pump. It's present on the apical membrane. Hydrogen is moved against concentration gradient with the utilization of ATP. And on the basolateral side of the parietal cell, we have chloride bicarbonate exchanger, which exchanges one chloride ion for one bicarbonate ion. This chloride ion diffuses into the gastric lumen, resulting in the formation of HCl, whereas this bicarbonate is being absorbed into the plasma, thereby increasing the alkalinity of the blood. So, with increase in the gastric acidity, there is a proportionate increase in the alkalinity of the blood, resulting in postprandial alkaline type. Now the HCl secretion part is over. We move on to the secretion and the release of pepsinogen. So we all know that the pepsinogen is getting released from a specialized cell called chief cell or peptic cell. Usually it is secreted in the proactive form, which is pepsinogen. These are usually stored in the zymogen granules of the cell until it is being stimulated. Whenever it is stimulated, it secretes pepsinogen. This pepsinogen is converted towards active form peptide by the action of HCl. This pepsin function is mainly to act on the dietary protein and digestion and form the products of protein digestion. Then we have surface neck cells and mucous neck cells. Okay. So this mucus secretion from these mucous cells, they are highly thick and viscid. I've told already, it forms a barrier between the gastric mucosa and the gastric content. So this is the gastric mucosal cells. This is the gastric content. And can you note down this yellow color? This is the mucus bicarbonate layer. It is almost a thick of about 1 millimeter or more than that sometimes. It is highly rich in bicarbonate, so it can neutralize the chemical component and forms a physical barrier, which will not allow the gastric mucosa to come in contact with the gastric acid content, thereby preventing its erosion and ulceration. This mucus secretion from the mucous neck cell and surface mucous cells, if we see, we have two types, soluble and insoluble mucus secretion. Mainly from the cardiac and the pylovic region, that is from the cardiac glands and the pylovic glands, we have mucus cells and mucus secretions from them. They are soluble in nature, but whereas insoluble mucus secretion is present all throughout the stomach, which mainly involve in the formation of mucus bicarbonate layer. Now, the alkaline acts as a gastric barrier, helps in buffering both physical and chemical function. Now, we move on to the factors which stimulates gastric acid secretion. So the main stimulus for gastric acid secretion, if you see, that can be a mechanical stimulus, that is distension of the stomach by the food. So there is gastric relaxation to accommodate the food border separate the stomach. So that can be a stimulus as such, which initiates a local vasovagal reflex, resulting in the stimulation of enteric nervous system. We know what is enteric nervous system. There are two important plexus which forms the enteric nervous system, myenteric and mesenteric. Okay, they get stimulated. Then apart from this, the products of digestion like carbohydrates, proteins, and lipids, mainly proteins helps in the stimulation of pepsinogen secretion. That we have to remember. Okay, so what happens after that? This EMS and vasovagal reflexes stimulate the release of acetylcholine from the nerve ending, which can act on the G cells which is present in the pyloric antra. So what is the function of G cell? We all know from the GI hormone class that it releases a hormone called gastric, which is a stimulator of HCl secretion. So it gets stimulated, it releases gastric. So this vagus nerve can also act on the ECL cell. So this ECL cell is nothing but enterochromaffin-like cell which releases histamine. I've told you this histamine is a potent stimulator of HCl. How? This histamine, it acts on the G cell to release gastrin, can act on the parietal cell. We have H+ receptors, H2 receptor, especially on the parietal cell. This histamine binds with its receptor, especially the H2 receptor on the parietal cell, and stimulate the release of HCl from them. So this is how it is being stimulated. So from this picture, you can note down. So this is the G cell, this is the ECL cell, G cell secreting gastrin, ECL cell secreting histamine, this is the parietal cell, this is the nerve ending releasing acetylcholine. Okay. So whenever the G cell is stimulated, it releases gastrin, it enters the blood circulation, it can reach the parietal cell, stimulate. These G cell releases gastrin, can go and act on the ECL cell, stimulate them to release histamine, which can act on the parietal cell, release histamine. Neuronal ending can place acetylcholine. This acetylcholine can go and act on the PCL or it can directly act on the parietal cell and causes the stimulation of HCl secretion. From this picture, you can also note down one other thing that the secretion of gastrin from the G cell has to come and act on the parietal cell through the blood circulation. Whereas this ECL cell releasing histamine can directly act on the parietal cell, not even entering the blood circulation because they are present one near each other. It can act in a paracrine fashion, directly acting on the receptor present on the parietal cell, especially the H2 receptors. It binds with its receptor and stimulates the release of HCl. So these are the factors which helps the stimulation of HCl secretion. Now we shall move on to the factors which inhibits gastric acid secretion. So mainly the hormone which is present in the pyloric antrum. That is, we have a specialized cell called D cell in the pyloric antrum which releases somatostatin. This somatostatin is a local GI hormone which has an inhibitory effect on G cell. So we already know G cell is involved in the release of gastrin, which is a potent stimulator of gastric secretion. What happens whenever the somatostatin goes up, inhibits the activity of G cell function will be decreased. So whenever the G cell action or the activity is being decreased and reduced, the release of gastrin from the G cell is also altered. That is, it is being decreased. So when the gastrin level, which is getting released from the G cell is reduced, the amount of gastrin reaching the parietal cell will also be reduced. So the stimulant for gastric secretion decreases. So the HCl secretion finally goes down. So apart from somatostatin, we also have the influence of few other hormones like cholecystokinin, secretin, GIP, gastric inhibitory polypeptide. So these hormones gets released from the intestine. Somatostatin, we have seen that it is getting released from the D cells of the pyloric antrum. Whereas this secretin and CCK, they all get released from the upper part of the small intestine. So this is the cycle you can note of. Instead of entering the stomach, it is getting mixed and churned by the acidic content of the stomach. Then when gastrin is released, it gets mixed with them, then it gets emptied into the intestine through the pyloric sphincter. So whenever it is getting emptied, first the D cells gets activated, somatostatin inhibiting gastrin. Then whenever it enters the upper part of the small intestine, the first part, the small intestine, what happens? It stimulates the release of hormones, CCK, secretin from I cells and S cells of the upper small intestine. So the function of these hormones, if you see, CCK and secretin, they stimulate biliary and the pancreatic secretion. So pancreatic secretion, CCK, it stimulates the enzymatic portion of the pancreatic secretion. That is the secretin, it stimulates the aqueous component or the serous part of the pancreatic secretion. That is rich in water and electrolytes, especially bicarbonate. So what happens whenever the food reaches here? The bicarbonate rich aqueous component will go and utilize the acidic chyme which is entering the intestine from the stomach. So it has to utilize the acidic content, or else what happens? These acidic content can erode the intestinal mucosa. That is why it is highly needed and it is released in response to the presence of acidic chyme in the upper part of the small intestine. So what is the stimulus for secretin then? It is the presence of acidic chyme in the upper small intestine. Then function of CCK is the enzymatic portion of the pancreas is being increased. So how will it get stimulated then? Products of carbohydrates, products of protein and products of lipid digestion in the gastric emptied chyme stimulates the CCK release. Then this CCK can also act on the contraction of the gallbladder resulting in the expulsion of bile, which is highly needed for the emulsification of fat. So whenever the chyme content released from the stomach is rich in lipid, stimulates the contraction of gallbladder, resulting in the expulsion of bile into the intestine. So once it is done, what it will do? It will neutralize the content, prevents the gastric mucosa, it aids in digestion. Apart from that, these hormones can also inhibit the action of gastrin Then it can also decrease the gastric secretion and gastric emptying at the same time. This is how the gastric secretion, that is the HCl secretion from the stomach is decreased and its emptying into the intestine is also delayed. So, overview, if you check out stimulating factors, you have to think about acetylcholine, histamine and gastrin mainly. So, this G cells to the blood circulation, ECL in a paracrine fashion and acetylcholine from the neuronal entity, both vagal and the ENS. Then we have the factors inhibiting gastric secretion. We have to think about somatostatin from the D cell, CCK, secretin, and GIP from the upper pituitary glands. Phases of gastric secretion. We will, if you see, we have already discussed about the different phases of secretion in different parts of the GI tract. Here, just to remember about, you will always think, see, cephalic phase, gastric phase, and intestinal phase. You know what is cephalic, gastric, and intestinal. The gastric secretion is high during the gastric phase when compared to the cephalic and the intestinal. So, and it is mediated mainly by the vagal nerve ending releasing the acetylcholine. And apart from neuron, there is also hormonal effect. We know that is gastrin, histamine, and acetylcholine acting on the G cell, acting on the ECL cell, promoting HCl release. Thank you. This is about gastric secretion. We can just revise it in a nutshell about gastric secretion, that the cell from which it is being secreted is the parietal cell, which we have to remember. So in resting state, the canalicular pit is smaller, but in active stage, if you see, the tubules and vesicles get incorporated in the canaliculus and the surface available for the secretion is increased. Then this hydrogen has to move against the conservation gradient. Hydrolysis of ATP occurs. This potassium moving in exchange for hydrogen is being recycled again back into the gastric lumen through potassium channels. For every hydrogen moving out of the cell into the lumen, there is one bicarbonate ion moving into the plasma in exchange for chloride to chloride bicarbonate exchange, thereby creating postprandial alkaline type. The concentration of sodium and potassium inside the cell is maintained by sodium potassium ATPase pump on the basolateral side. That we'll have to remember. So these two pumps we'll have to remember. So hydrogen potassium ATPase pump on the apical side and chloride bicarbonate exchanger on the basal side. Then pepsinogen from cheek cell, mucus secretion from surface, and mucus neck cell. In factor stimulating, gastrin, histamine, acetylcholine, inhibiting, somatostatin, CCK, secretin, and GIP. That's it for today's class. Thank you so much for listening.